Revolutionary AI advances are reshaping gene editing, tackling one of its most daunting obstacles, predicting DNA repair after a cut. This new frontier could redefine the safety and effectiveness of genetic therapies, but here's where it gets controversial. How accurate can AI predictions truly be across diverse biological systems? At the forefront of this advancement, a team of researchers led by experts from the University of Zurich has created an AI-powered tool named Pythia that vastly improves the precision of CRISPR gene editing. Pythia's breakthrough lies in its ability to forecast how DNA repairs itself following the disruptive cuts made by CRISPR technology. A critical step, since the cell's natural repair mechanisms are prone to errors that may damage important genes or produce unintended mutations. The collaborative effort between the University of Zurich, Ghent University in Belgium, and ETH Zurich. By using AI algorithms, Pythia analyzes massive datasets to uncover patterns in DNA repair, challenging the former notion that such processes are merely random occurrences. As Thomas Naert, the lead scientist who initially developed this at Zurich and is now a postdoctoral researcher at Ghent University, explains, "We designed tiny DNA repair templates that act like molecular glue, guiding the cell to make exact genetic corrections. These templates improve the precision of edits by steering cellular repair mechanisms toward desired outcomes." Their work extended from controlled laboratory human cell cultures to living organisms. Notably, they demonstrated successful gene editing in Xenopus, a tropical frog frequently used in biological research, and even achieved precise DNA modifications in the brain cells of live mice. These accomplishments highlight Pythia's versatility and potential for complex tissues, including organs like the brain, where cells rarely divide, historically a tough challenge for gene therapies. Understanding the predictability of DNA repair is the cornerstone of this project. Naert emphasizes DNA repair isn't arbitrary, it follows specific, learnable patterns. This insight allowed the team to simulate millions of possible genetic outcomes using machine learning to answer a pivotal question: what's the most efficient pathway for introducing exactly the change we want, given the cell's repair tendencies? But Pythia's applications are even broader. Beyond editing single base pairs or inserting entire genes, this approach enables fluorescent labeling of individual proteins. This technique provides scientists with a powerful window into visualizing protein behaviors directly within healthy and diseased tissues, offering richer insights into cellular functions and disease progression, something that can supercharge research and drug development. Søren Leinkamp, a senior author of the study and professor at both UZ and ETH Zurich, draws an intriguing analogy. Just as meteorologists rely on AI to predict the often unpredictable weather, we now use it to anticipate cellular responses to genetic editing. Such foresight is vital to ensure that these interventions are not only effective, but safe and trustworthy for clinical use. What truly makes Pythia groundbreaking is the seamless integration of large-scale AI predictive modeling with experimental biology, from petri dishes to living animals. This robust feedback loop between computational predictions and real-world validations brings us closer to tailored gene therapies that minimize risks and maximize benefits. This research opens exciting doors for future treatment options in genetic and neurological disorders, many of which currently lack effective cures. However, one might ask, given the complexity of human biology, can AI models like Pythia entirely capture the unpredictability of DNA repair in every tissue and individual? Could over-reliance on AI predictions potentially overlook rare but critical exceptions that impact patient safety? These questions invite vibrant discussion in the scientific community and among you, the readers. Do you believe AI-driven precision is the future of gene therapy? Or should we maintain caution amid the complexity of life's code? Share your thoughts and join the conversation below, because when it comes to editing life itself, there's always more to explore.